Genome organization and synteny. Tailed phages have linear double-stranded genome. We learned last semester that the left arm, in general, codes structural genes. The right arm encodes replication and early lytic genes. And then temperate phages, immunity, integrase, and ATP sites are usually located in the center of the genome. Although phage genomes are highly diverse and mosaic, as these three phage genomes are, they often share similar genome organizations. Here are the left arms of genomes from cluster F, G, and K, and each encoding the structural genes. They don't share very many gene families, yet certain gene functions are found in the same genome location. For example, the terminase genes are typically found at the beginning or nearly the beginning of the left arm. The terminase genes are nearly always followed by the portal protein genes. This is called, where the gene order is conserved on a chromosome or genome, synteny. The genome map of the Mycobacteriophage tweety shows the canonical order of structural genes on the left arm of the genome. The first genes are the small and large subunit terminases, followed by the portal protein and the capsid maturation protease, the scaffold protein, the major capsid protein, head-to-tail connector proteins, followed by genes involved in assembly of the tail. After the minor tail proteins are genes that are usually involved in lysis. The right arm of the genome encodes genes expressed during early lytic replication. These are usually transcription factors and genes involved in DNA metabolism, including DNA methylases. Although not seen in the tweety genome, there are often genes that encode DNA polymerases, helicases, and primases. Temperate phages typically encode genes involved in lysogeny establishment and maintenance in the center of the genome. This is true for tweety, and we see here an integrase and adjacently the ATP site. Also note a reversed-oriented immunity repressor and forward-oriented CRO gene, an arrangement similar to that of lambda phage. This is a typical arrangement of lysogeny genes, and we can use this arrangement as a model to predict gene functions in other temperate phages. Take a moment to examine the genome organization of your phage genome in Famrator. It will be helpful to align your phage genome to other members of the same cluster. Does your phage genome have a typical organization of genes on the left and right arm? Make note of any genes that occur in unusual locations. Do you have a temperate phage, and are there lysogeny genes organized like lambda, or more like L5, the cluster A2 phage we studied in Journal Club? Why do we care about genome organization and synteny? We can use our knowledge of phage genome organization and synteny as a tool and evidence to predict novel phage gene functions. For example, if we just annotated and predicted the function of the phage tape measure gene, what is the most likely function of the gene that follows? This is something we'll talk more about in class today. Your tasks to prepare for class are to learn the function of each of the phage structural proteins. If you are rusty, see the anatomy of a phage video. Learn the basic organization of the phage genome, especially for the type of phage you are annotating. And learn about genome ends and genome packaging. Focus on COS end packaging mechanism.